Hi, awesome disease and public health students. We're going to do a short little hands-on demonstration to explore how two sort of cellular check mechanisms can prevent cancer during cell division, specifically tumor suppressor genes and proto-oncogenes. You're going to need a flat surface on a desk to film on, and you'll need either a stand or somebody to help you film. You're going to need either string or yarn or thread or floss is what I'm going to use to represent your nucleus and your cell membrane. So you should have sort of two smaller circles to represent your nuclei. Here are my two pre-tied floss strings, and you'll need two other ones that are twice as large. So I usually make my large ones about twice the length of a standard ruler, and the shorter ones for the nucleus about the length of a normal ruler. Again, that's about 30 centimeters on a normal ruler. So you'll also need to have something to represent the DNA. You'll need 4 different items, and you can alternate them. So we want the normal DNA, so you could use something like a leaf, and then the mutated leaf would be one that is chopped in half. You could use something like a piece of food, like a chip or a pretzel. Here's my normal DNA pretzel and my mutated DNA pretzel. You could use a paper clip or something, and then a bent paper clip, so that's my normal, my mutated DNA. There's lots of different options you could use, so whatever kind of lines up to represent those. And then you'll need to make 3 Post-its or 3 sheets of paper, one that just says check on it to represent the cellular division check mechanism. And we're first going to explore tumor suppressor genes, which stop cell division if things aren't looking right in the DNA category, and proto-oncogenes, which are more like the go mechanism, which again will not signal unless things look correct. And so I'm going to play through how these should normally work, and then you're going to explain to me what would happen if something was wrong with the tumor suppressor gene or the proto-oncogene. So to start off, throw down your nucleus, your cell membrane, and a representation of your DNA. I'm going to go ahead and use the paper clips. So we've got a mutated piece of DNA in here, okay? There are numerous checks that happen during cell division, and in this one I'm going to explore what's going on with the tumor suppressor gene. If this was mutated DNA, the tumor suppressor gene is activated and it stops cell division. Nothing happens until that DNA, or the mutation inside of it, has been corrected. Now let's say there is normal DNA there. The tumor suppressor gene is switched off, and so what happens is then we end up with a second copy of that normal DNA inside a second nucleus, and then the whole thing pulls apart to form 2 cells, like that. That's normal cell division, and we've ended up with 2 cells that have the normal DNA inside of them. So I would like you to, in your video, show me what would happen if the tumor suppressor gene was not functioning properly. Okay, second video, we're going to go back to having this 1 cell. This time we're going to explore the role of the proto-oncogenes. Again, we're going to start off with mutated DNA in the center, and this is just another mechanism. I think these two can work in conjunction as well, but we're just going to look at them separately. So once again, a check would occur in cell division. This time we're going to look at the role of the proto-oncogene, but since this DNA is mutated, it's actually not going to be present. This will only give the go signal if the DNA is not mutated, right? If it's ready to go, if it's normal. And so once again, nothing will happen unless that mutated DNA is turned into normal DNA again, no mutation. Now the proto-oncogene says go, and once again we end up with 2 nuclei that have a copy of that unmutated normal DNA, and then they split into 2 separate cells, each containing that normal DNA. So again, in your video, I'd like you to quickly show me what happens if this proto-oncogene is not functioning properly, which usually means that it is just stuck in the go position no matter what happens. Have fun. Reach out to me if you're confused about what to do. Thanks.